pleasure to introduce Victor Velculescu, MD, PhD. Victor got his bachelor's degree at Stanford University, went to that little-known institution, Johns Hopkins, for his MD, and got a PhD in human genetics and molecular biology at Hopkins. He rose from assistant professor to professor in a few years, and he's currently professor of oncology and has many other great titles there. I want to tell you about this man in a different way. He's been awarded so many honors already. Rockwell International Achievement Award in Science and Mathematics, the Firestone Medal for Excellence in Research from Stanford. I didn't know he was royalty, but the Order of Merit, Cavalier, President of Romania. Brilliant 10 scientists, Young Scientist of the Year by Popular Science, a Pew Scholar, the Judson Dayland Award of the American Philosophical Society for achievements in identification of diagnostic and therapeutic targets through genome analysis of human cancers, the Paul Marks Prize for creation and application of novel technologies to detect alterations in human cancer. He has incredible publications. I won't go through them all, obviously, but he developed many of the techniques, nearly all of the techniques we use, called SAGE, Serial Analysis of Gene Expression, prevalence of somatic mutations in colon cancer. That's why we wanted him to work on pancreas cancer. The RAS genes, which are so important in melanoma. Everybody uses this today, the digital karyotyping, a breakthrough technology. High frequency of PI3 kinase mutations. Mutations in colon cancer and brain cancer. Epigenetic changes, I talked about manicuring the DNA, that comes from Victor's work. Profiling of cancers for diagnosis. Serial assessment of human tumor burdens in mice by analyzing circulating DNA. Core signaling pathways. The first description of mutations in glioblastoma, which is as bad a cancer as pancreas cancer. I could go on and on. Just let you know this man, obviously you can tell, is very accomplished, and his most recent work is in predictive capacity of personal genome sequencing. So without further ado, I'd like to introduce Dr. Victor Velculescu, who's going to speak to us on pancreas cancer and personalized genome analyses. Victor.
Thank you very much, Dr. Von Hoff. It's always a pleasure to be with you at a meeting. I've been to several of his, and it's a pleasure to be here today. It's actually really inspiring for someone like me who is in research and is working on the genetics of this disease to see not only those that could benefit from it, but the support and the large number of individuals that are here today that are really trying to make a difference in this disease. So I've been inspired, actually, sitting in and being a participant here today. So I hope I can share with you today some of the work that we've been doing and have been doing actually part of a large collaborative effort. This is work in part by Johns Hopkins, in part by TGen, and in a way I'm honored to be really part of Dr. Von Hoff's team, really from afar, doing work on the genomic analyses of pancreatic and other human cancers. And what I'd like to do today is to tell you some of the ways in which this type of analyses have been done. There's really been a tremendous explosion in technology that have allowed these to be possible, the lessons that have come out of these, and the implications that they have going forward for future diagnostic and therapeutic approaches. So the work I'm going to tell you about today rests on the idea, which has now been documented by about 20 years of research, that cancer is a genetic disease. It's a disease where some of the bases or some of the letters in the genetic blueprint in one of the cells in our bodies ends up being changed. And these changes can be single letter changes or they can be larger changes. And if the genome were like an encyclopedia of 3 billion letters, they could be little typos, or they can be essentially new chapters that are added, or chapters that are deleted, or chapters that are rearranged. And this mix-up of information makes these cells go awry and makes them grow abnormally. And with time, the accumulation of these mutations lead to cancer. And so for a long time, trying to understand these genetic events was very, very difficult. People literally spent decades trying to find a few of these genes, and a lot of progress was made, but it was really very slow and very painstaking. And what's happened recently is that there's been a revolution in the ability to analyze these genomes in cancers and to find the entire genetic code, not just of some tumor specimen in a laboratory, but of patients that are actually in the course of being treated or in the course of a clinical trial. So the combination of new technologies and cancer genetics now really allows, I think, for really a paradigm shift in the way we've been thinking about cancer, where this information can be used in a very personalized way, in a very precise way, to allow one to think about diagnosing and treating patients differently. And the analysis not only of tumors, but of the blood can provide a very powerful diagnostic test for patients with cancer, because this DNA that's present in the blood is really contains both normal DNA but also mutated DNA. And the mutated DNA only is representative of the cancer and is a very specific marker of this disease. And so we've been working for many years on ways to detect this mutated DNA for reasons of early diagnosis and detection of cancer. But the challenge has been, how do you identify these small number of mutated molecules and the large amount of normal DNA that's there? And so we've developed an approach that was just published actually last week in Science Translational Medicine. And what this approach does is it allows sequencing of DNA that's free-floating in the blood using these new sequencing technologies to identify the type of structural alterations or big chromosomal changes, the change of chapters in the encyclopedia of our genetic code, just by looking in the blood of individual patients. And so what this work has shown is that if you look in 10 patients that have either colorectal or breast cancer, you see gains and losses of chromosomes. This is, again, in the blood of these patients, while in healthy individuals, you don't see any of these changes. The other thing that it's shown is that all of these individuals, when you look in their blood, you can find what are called rearrangements, so fusion of two pieces of DNA from part of the genome that are normally never together. This is a pictorial of these rearrangements, starting with chromosome 1 all the way to chromosome 22, and these sort of arches show this rearrangements or joining of pieces of DNA. Again, all the patients had this. None of the healthy individuals have this, and all cases of cancer that we've looked at to date have these rearrangements present in the cancers. But now, for the first time, really using these technologies, we can identify these very precise junctions in the blood of these individuals. So how is this useful? Well, I think it'll be useful in at least several ways. One is that we really need better ways to monitor patients during therapy. So this is an example of a patient that was a colorectal cancer patient that had a removal of the colon cancer, and you can see here using this molecular marker of this rearrangement in the blood that their cancer obviously did better. It went down, but it didn't go to zero, suggesting they still had cancer there. It went back up, and then over a much longer period of time of therapy and further surgery, the levels went down almost to zero. Unfortunately, it's not zero because the patient still had a remaining lesion in the remaining portion of their liver. So this molecular alteration allowed for a real-time molecular evaluation of how the patients are doing as we get new therapies, as we want to see whether patients are responding or not responding to a particular therapy. Having this sort of real-time response, I think, will be crucial in the future. A second way in which this type of methodology will be useful is answering the question that every patient has after surgery of whether or not they've been cured by the surgery. Every patient wants to know that answer, but no surgeon can definitively tell you that. And so many patients, for example, are treated unnecessarily when they're early stage patients or when they're late stage patients, even though they've been cured. And many early stage patients that would have been cured by the tumor or by the surgery, unfortunately, are not cured, and their tumor recurs, and they're not treated, even though they could receive more intensive therapy. So this is an example in colorectal cancer where this type of approach was used, and when no mutations or no alterations are detected in the plasma, all of the patients did wonderfully well and none recurred, while those patients that had mutations in the plasma right after surgery, unfortunately, all recurred and most of them succumbed to their disease. So if we could have better ways of telling patients after surgery whether or not they still have residual disease, whether the surgery worked, we could know what better therapies to give them right at that time. The third way in which this will be helpful is to tell patients immediately when you can't look at the tumor whether they have a genetic alteration that can be targetable. This is an example of a HER2/neu amplification that's present directly or detectable directly in the plasma of this patient. The HER2/neu is the target of Herceptin, a therapy used in breast cancer. In this case, it was in a patient with colon cancer. And that's very useful information in cases where you can't get the therapy. And finally, or actually not finally, penultimately, many therapies these days that are targeted get resistance. So the therapy works for some time, but ultimately the tumor comes back. Can we tell whether the patient is getting recurrence of the disease prior to them showing clinical signs of that? So looking for these mutations in the plasma, example here of KRAS in the blood, identifies these mutations in the plasma prior to clinical detection of the cancer. That's a very important aspect and could tell doctors to switch therapies sooner. And finally, something that we've heard about today a lot of is early detection. And so we're very interested in applying this technology to early detection. The amount of sequencing that we can do currently can detect levels of this mutation somewhere between 0.1 and 1% at a in a cost-efficient way. But the decrease of sequencing costs that's been happening over the last number of years really makes us hopeful that we can detect levels even lower, which we know we can, but we just have to do more sequencing and it has to become affordable in the near future. And so this type of approach, I think, will be useful in the near future for early detection and for finding these cancers early, intervening prior to them becoming a very late-stage disease. So I've talked today about really the very exciting transition that's occurred in taking this work that's been done in the laboratory for the last 2 decades and really being able to have it used on patients in an everyday manner. And this, I think, is going to continue fueling the discovery of new biology and therapeutic targets. It'll allow us to use these genomic analyses in clinical trials for very specific and precision intervention. It'll allow us to really begin helping patients with these types of changes, allowing us to detect disease and to monitor disease in patients like I've shown you, to detect and overcome therapeutic resistance, and with a long-term goal of early detection and early intervention. And this is important really for all cancers, but I want to just highlight that it's incredibly important in pancreatic cancer, and I'm stealing a phrase here for Dr. Von Hoff of the relentless pursuit to achieve these goals in pancreatic cancer. So with that, I wanted to just thank all the folks that have made this work possible, the trainees and my colleagues at Johns Hopkins, and the folks at TGen and the other institutions for the Stand Up to Cancer. Thanks very much.